There we go. The second one? All right, it's... Oh, 3 people are watching. 3 people are watching. Hey. It does look a little bit stretchy, but not terribly stretchy. What is the resolution of that thing? Yeah. Yeah, it's actually because it is a lot. Yes. So, try something. Yeah, and it's actually also what? We'll presumably pick it up, kind of. It's a best effort kind of situation.
Yeah, I know. RDF's 3 days. I didn't find it.
It's been like a year and a Oh my god. Like you like do one, but it's like possibly it's hard.
I feel like this, like yeah, I know. It's nice to see you.
Those are like heavy. Wow. Yeah, those are my water. Mine weighs like double. That's where it stands. Oh my god.
We're happy.
So I guess it's time to get started. Welcome everybody. This is the public defense for Stephanie first. And I would first like to take a moment to welcome her family and her friends who support her today. That is Lori Nunley, her mother, and then David Hurst, her father, her grandmother, Judy Wilkinson, welcome. And then we have her younger sister, Laura, welcome. And then we also have the family of her husband, Luca is here, with his parents, Alice and Ed. Welcome. It's great to have you all here and celebrate this day together with us. I remember it quite distinctly because I hadn't joined faculty that long. I think it was about 2 years or so. And I got a phone call from Albert. And he said, no, an email, or can you interview this young woman? She applied to our graduate program, but she is in Germany. And he probably thought because she's in Germany, I could very well interview her. And so we had a phone interview at the time. And you know, we were talking about graduate school and all of her qualifications, which I should mention at the time, Stephanie had done an internship in Germany with a Fulbright fellowship, I believe. And there she already had done research in computation, actually. And so, and then I got back to her back and said, yeah, sure, I think she will be successful in our graduate program. And this is when Stephanie joined the CPD program and later also joined my group. And today, some, I don't know, 6 years later or so, we are here to listen to Stephanie presenting her PhD thesis. And one thing that I should make clear to everybody, this is certainly not lightweight. So she did a massive amount of work, and she already published out of her PhD thesis 4 papers, and then there is one more that is submitted, and one more that we will have to click submit, I guess. And so this is a total of 6 papers, and on all of these 6 papers, she is either the first author or a co-first author, which really shows that on all of these 6 projects, she really was critical to the success of the scientific project I'm here. You know, and this is a very, very proud summary for a PhD advisor, a student who leaves the lab with 6 first or co-first authorships. That's certainly 100% plus X done. So I'm very proud of Stephanie for that, but also for a number of other reasons that you are aware of. You are aware that Stephanie is legally blind, so she counts as underrepresented minority. And I don't mention that today because I think that's special. I mention it today because the most amazing thing to me was that I had forgotten about that after about 3 or 4 weeks. I treated her as every other student. She needed, I told her, remind me if there's any special thing I need to do for you, because I will forget. And so she went through this graduate program as any other student in the program. She had the same hills to climb, the same hardships to fight that every graduate student experiences, and she did a wonderful job. And I don't remember a moment when there was anything that we needed to do special, other than the usual accommodations that you would expect, but anything where she took a shortcut because of this. And I think this is really true accomplishment of her that speaks to her moral fiber. There were certainly things, that in every graduate student's career, and in hers for sure, there were moments in time that were really tough, and she really had to fight and dig deep. And sometimes people had to back her arm and say, you're just gonna have to do it. There's just no way around it. And we discussed sometimes about whether or not it had to be done, but in the end, she was off to do it, and she did it, and this is why we are here today. So I'm very proud of having trained Stephanie for many different reasons, but I think this is really one of the experiences that I will remember of Stephanie for the rest of my life, because she's a very special person, and despite you know, all of the extra work she had to do, she did a great job and lived up to the expectations entirely. So I very much look forward to her presentation today, where she will convince us that she is a PhD. And after the public defense, there will be a private defense where, again, the committee who is here will have the opportunity to ask her questions until she says, I don't know. And provided that we get to the point where she says, I don't know, and the private session is successful, then we're going to have a reception, and I would expect that this might be around 3:30, 3:45 or so on the fifth floor. And any other outcomes we don't even discuss. Stephanie, all yours.
I wasn't sure if Jens was going to show embarrassing slides of me or not. Okay, so thank you, Ian, for that very kind introduction. I already started tearing up before the thing starts. So today I'm really glad you're all here because I'm excited to tell you about the research that I've been doing in the minor lab for the past 6 years. And this research has to do with developing tools for computational modeling that combines, thank you. I don't know how to work the that combines computational and experimental methods. So I'm going to cover a lot of ground today, and so this is just an outline to kind of walk you through. Basically, I'm going to give a relatively brief introduction concerning proteins and why they're important to us and why we choose to study them, and then also an introduction to de novo folding of proteins and protein structure prediction in Rosetta with EPR restraints, and how we can apply that to membrane protein structure determination. And finally, sum it up and acknowledge that people who helped this happen. So, I thought, there are some people in the audience who might not be as familiar with the biochemistry or the point of studying proteins, so I thought I'd start with that. And I thought I would give an example of why proteins are important in our everyday lives using a case study where the subject of the case study is myself. And Jens already alluded to the fact that I'm legally blind, which was kind of took me by surprise. So as you might have noticed, I'm albino, and this basically means that, instead of looking like these Korean ladies here, I look like this, which has its certain disadvantages and advantages. And, specifically I have a type of albinism called oculocutaneous albinism, or OCA1A, which has several symptoms, including severe photophobia or light sensitivity, severe nystagmus or involuntary movement of the eyes, and overall low resolution vision. And that has to do with, low resolution vision has to do with the lack of a well-defined phobia on the retina. So if this is a normal retina, a person with a normal vision, and this is the retina from someone with OCA1A, you can see that here there's a dark spot where the cones, as in your rods and cones, are concentrated here. Whereas there's no dark spot here, there's no well-defined fovea. It's because the cones are spread out 3 to 4 times more than in a normal eye, and therefore you get low resolution vision. And what I mean by that is, for example, if you have 20/20 visual acuity or normal vision, then you might see text that looks like this. But if you're on the border of being legally blind, so 2200 vision, it might look something like this. And as the text gets smaller, you can see that it gets much more difficult to resolve. And so this is basically kind of a simulation of what someone who's legally blind would look like. It turns out that's actually my corrected vision with context. So, you know, what does this have to do with proteins? Most of you know, if not all of you, that our genes are made up of DNA. And for example, I'm showing here a map of the TYR gene, which is important for the albinism story. DNA can be kind of thought of as the instructions for building biological machines. So in this case, we have the TYR gene, which those instructions are transcribed into RNA, which then are translated into the three-dimensional protein tyrosinase. And tyrosinase is the biological machine that performs the first two steps of melanin synthesis in our melanocytes. And so you can imagine that if you're albino with OCA1A, you might have one or more of these mutations, which then leads to transcription into RNA of a different form based on those messed up instructions, which then leads to the production of a non-functioning tyrosinase enzyme. And because this molecular machine is not working anymore, then there isn't any melanin. And therefore, since I don't have any functioning tyrosinase, basically I'm really pale and have bad vision. But you know, this is all to drive home the point that, you know, protein malfunction can, or lack of a protein, or can have small effects, but they can also have very drastic effects. And so the idea is that if we can understand, behind our research is that if we can understand what these proteins look like and how they function, then we can design better drugs and therapeutics to treat all sorts of diseases. So not even just albinism, but cancer, Alzheimer's disease, and any number of things, because proteins are involved in pretty much everything in our bodies. And as a structural biologist, you know, we believe that the structure, the three-dimensional structure of a protein is highly correlated with how it works. And so there are a lot of advances being made in trying to figure out what these proteins look like, develop methods to figure out what these proteins look like. And this is what my research has to do with. In our cells, we have two main types of proteins. There are soluble proteins, which live out here in the cytosol in aqueous solution, and then you have membrane proteins, which live in or reside in the lipid bilayer. And here's a zoom, and the lipid bilayer surrounds cells and our cells and our organelles. And if you zoom in into a section of the lipid bilayer, you'll see that there are also subtypes of membrane proteins. There's lipid anchor proteins, peripheral membrane proteins, and integral membrane proteins, which span this entire layer. And when I talk about membrane proteins in this presentation, I'm actually pretty much always referring to integral membrane proteins, and more specifically, probably more often than not, alpha helical membrane proteins, which is just a specific type of structure. And you know, what's interesting with membrane proteins is that, you know, because of the environment in which they reside, they're kind of greasy or oily in nature, and we as structural biologists have to treat them quite differently than we do soluble proteins. My research is primarily focused on membrane proteins, although I will spend some time today talking about work that we performed on soluble protein, T-lysozyme. Membrane proteins also stand out because they're really popular drug targets. That is that many drugs and therapeutics, their mechanism of action has to do with how they interact with membrane proteins. And it turns out that more than half of FDA-approved drugs actually are classified as membrane proteins. And here's some subclasses. Over a quarter of FDA-approved drug targets are in the family of G-protein-coupled receptors, which include the histamine receptors and NMDA receptors. And then like approximately 3% of the drugs on the market target neurotransmitter sodium symporters, which include serotonin transporters and noradrenaline transporters. So these are just some common drugs that you guys might be familiar with. And so, we're really interested in characterizing this type of protein because of their prevalence in drug discovery. However, the problem is that there aren't very many membrane protein three-dimensional structures available to us compared to the number of soluble protein structures. In the protein data bank a couple of weeks ago, there was about 100,000 protein structures deposited overall, but less than 2% of those were of membrane proteins, and the majority of which were determined by X-ray crystallography. Although there also is a growing number that is being determined by NMR spectroscopy. So there are a lot of advances. This discrepancy between how many structures we have for soluble versus membrane proteins points to a technological challenges that arise that it's associated with membrane protein structure determination. For example, in X-ray crystallography, or for example, membrane proteins, they can be often difficult to express and purify and crystallize due to their greasy nature, and because they have to be studied in their native environment or in a lipid environment, then they can be difficult for structure determination with NMR spectroscopy because they're too large and it's hard to get a signal. So there's still room, despite the advances being made in these fields, there's still room for growth in developing new methods, different methods, such as computational protein structure prediction, which is where my research comes in. And as I was putting my thesis together, I was thinking about how we are approaching these problems, and I was kind of thought, this reminds me of the poem, The Road Not Taken by Robert Frost, where at the end of the poem he says, "Two roads diverge in the wood, and I took the one less traveled by, and that has made all the difference." And I always thought it was funny that, he never described this difference, or if it was a good or a bad difference, or just kind of was a difference, and I guess it was just up to the reader to figure it out. And I feel like, in our research, or in research in general, sometimes you just have to you're faced with a choice and how to approach a problem, and then you try to inform that choice, but in the end, sometimes you just choose something and go with it. And I feel like that's kind of what we did. And it definitely made all the difference. But anyway, philosophy aside, I would like to now dive into more details in terms of computational modeling. So there's a couple of ways to approach the computational modeling of proteins in general, not just membrane proteins. One is comparative modeling, in which your protein of interest, its structure is going to depend on the structure of an existing protein, an existing structure of a different protein, a related protein, and or an existing structure that has a similar fold to yours. The road less traveled, however, I think is probably ab initio or de novo folding, which you would want to do if you can't find a sequence homolog or closely related protein. And when you're doing de novo folding, you have to assess how well how energetically favorable your models are. And so there's two main ways of doing that. You can use physics-based potentials, which are, or sometimes they're called force fields, which depend on, or are based on Newton's laws of mechanics, and then you have knowledge-based potentials, which in that case we kind of look at nature and see what kind of things are commonly seen in nature, and think maybe Mother Nature knew best, and we will just assume. What we would call scoring or energetic assessment of our models. They also apply to comparative modeling. But today, going to talk about de novo folding, or folding from the primary sequence or amino acid sequence of a protein with knowledge-based potentials. And the tool that I use to do this is called Rosetta, or the framework that I worked in is called Rosetta. And Rosetta is able to rapidly sample secondary and tertiary structure information via its fragment-based assembly protocol. And that sounds really jargony, but I'm going to just explain it. What it means is if you have a primary sequence of your protein, so, proteins are essentially long chains of amino acids, each with their own chemical properties. And if you take the primary sequence of your protein of interest, and then in the Rosetta protocol, we'll divide it up into 9 and also 3 amino acid sequence windows, indicated by these blue lines here. And then, in our knowledge-based approach, what we do is we query the protein data bank, which is just this large database of proteins of non-structure, for sequences that match up with this sequence window, and we collect the 3-dimensional coordinates for those sequences. And so these sequences, these peptide fragments can come from a lot of different proteins in the PDB. You can imagine there would be a lot of different combinations of fragments for this peptide. And we do this over the entire primary sequence of the protein and collect all of the coordinates into what we call a fragment library. And then those fragments are used to build up a protein model from an extended chain. And I have a movie, and I hope it works. Many of you have already seen this movie before. It's an example of de novo folding of a small protein called ubiquitin in Rosetta. And this is the traditional de novo folding algorithm in Rosetta. And let's see if it works. Yay! Okay, so in gray is the crystal structure of ubiquitin and the experimentally determined structure. And rainbow is the model that we're building in Rosetta. And as you can see, we take those fragments from the fragment library I just described and insert the coordinates. We pick one randomly, insert the coordinates onto this extended chain, and then measure the energy, the energetic favorability of that conformation, resulting conformation, using Rosetta's knowledge-based potentials and scoring function for soluble proteins. And if it was a good move, then we keep that move and then choose another fragment and do it again. And if it was not so good of a move, if the energy of the model went up, then we throw it out and choose another one and repeat the process thousands and thousands of times. And you saw at first that the secondary structure of the protein kind of collapsed very quickly, and that was easy to do with the 9-mer fragment insertions. And then in the end, you get more finer details with the 3-mer fragment insertions. And finally, after the model converges, you would hope that you get an accurate structure that matches up model that matches up pretty well to the experimental structure, which we do in this case. The thing is, that was folding of a very small, relatively easy protein. Sometimes, though, we want to model larger, more complex proteins, and that might require the use of experimental data. And you might ask, why are we interested in combining these modeling? That's pretty cool. Why do we even combine it with experimental data? That's because conformational space that a protein can sample is really, really big. Just imagine that this is practically infinite. And we currently don't have enough computational resources to brute force our way through that conformational search space. So what we do in Rosetta is we narrow that search space by applying the local sequence bias using the peptide fragments, and also energy evaluation with non-local interactions using the knowledge-based potentials, which is fine for folding ubiquitin and maybe some other smaller proteins. But for more complicated proteins or membrane proteins, often it's not quite enough. You can't possibly sample enough with your computational resources. So then that's when the experimental restraints would come in, experimental information, and come in to inform your modeling. And this experimental information can come from different types of experiments. So it comes from nuclear magnetic resonance spectroscopy, or NMR, SAX, which is small angle X-ray scattering, and in my case, electron paramagnetic resonance spectroscopy, or EPR. And so, by combining all three of these methods and making sure that your models comply or satisfy these experimental restraints, hopefully you can come to a model or an ensemble of models that is what we might say is native-like, is that biologically correct form. So the question is, how do we actually get those restraints, or in my case, how do your collaborators get those restraints? What we want to use, well, what I wanted to use in my project was distance information from site-directed spin labeling electron paramagnetic resonance, or SPSL EPR. And SPSL EPR, it has certain advantages that I'll describe in a second. It's kind of labor-intensive to perform one experiment to get one distance, interresident distance, however. You have to mutate out native cysteines in your protein, mutate in cysteines onto the residues that you're interested in measuring the distance, and then spin label it, or attach a paramagnetic probe, essentially. Here's an example of a commonly used spin label methane thiosulfonate, or MTS. And once you spin labeled your protein and make sure it's functional, once you expose it to an external magnetic field, the resonance between the two unpaired electrons can be detected and converted into a distance probability distribution, which then that distance, is going to be measured in angstroms, and you can say, okay, between residue 1 and residue 12, those two residues are probably about 20 angstroms apart. And it just depends on the protein. You can do this multiple times. We perform this experiment multiple times to get multiple distances and use those restraints to inform your modeling. As I mentioned, EPR has certain strengths over other more traditional structural biological techniques such as NMR spectroscopy and X-ray crystallography. Namely, there is no crystallization required with EPR spectroscopy, and there are no size constraints. You don't have to worry about signal to noise ratio as much as you do in NMR. Also, importantly, especially for membrane proteins, is that the protein can be studied in its native environment. So, like I said earlier, membrane proteins live in kind of a greasy lipid environment, and so that is really important that you can maintain that environment when you're studying the structure. Also, you don't need very much of the protein, which, as I said earlier, is nice because it would be nice because sometimes membrane proteins are difficult to work with and purify. So, but like with any other method, EPR definitely has its weaknesses. I mentioned that it's a low throughput technique, so therefore you get sparse data sets, and that can be a bit challenging to gather up enough data to constrain our conformational search space. Also, any time you make mutation into the protein and spin label it, you want to make sure that that protein is still biologically active, it still functions the way it's supposed to. Also, the spin label, you have to remember during modeling that when we're turning these data into structural restraints for modeling, that the distances obtained from EPR are not distances on the protein backbone, but rather between two spin labels, which are attached to a spin label linking arm. And so, there are disadvantages, and what we were hoping to do in our lab is to combine the strengths of EPR with the strengths of computational modeling to be able to get to determine protein structures in atomic detail. And so the way that the method that we used when we were coming up with this was the tool is called Rosetta EPR. And this is the overall protocol that I used in my first paper testing Rosetta EPR on T4 lysozyme, which is small soluble protein. And I should say that, I talked a lot about membrane proteins, but we developed Rosetta EPR first on soluble proteins kind of as a first step towards getting to working on methods for membrane protein structure determination. So this is kind of like getting our feet wet, and then later on I'll talk about membrane protein structure determination. Anyway, so the first step for developing Rosetta EPR was that we needed to map the spin label distances obtained from EPR onto the protein backbone. And we do this using a motion on a cone model, which I'll explain in a second. And we wanted to actually develop a knowledge base potential for EPR distance data, which we use based on cone model statistics. And then after doing that, we combined that knowledge base potential with other Rosetta scoring terms and other terms in the Rosetta energy function, and then optimize the weight of that scoring term. And then after the development phase, we moved into the structure prediction phase for T4 lysozyme, where we folded 500,000 models of T4 lysozyme with this EPR, new EPR knowledge base potential. Excuse me. And then filtered out for models that were, energetically favorable and also complied with the 25 experimental restraints that we provided, and then filtered those, and then went through multiple rounds of all-atom refinement in Rosetta before assuming that the last model, the lowest scoring model, was our best guess as to the actual structure of the protein, and then we compare that to the actual experimental structure of the protein. As I mentioned here, we wanted to map the spin label distances to protein backbone distances. And the way that we do that is with what we call the motion on a cone model, or a cone model for short. The cone model is based on a popular spin label called methylsulfonate, which you might have seen earlier, which has a beta carbon to electron probe distance of 8.5 angstroms. And one way to model this spin label is to actually do it explicitly, where you model all the atoms. But for de novo folding, that would probably be pretty slow. So we wanted to use an implicit model. And so we assume that in solution, the spin label would actually occupy the three-dimensional space of a right-angle cone, where the distance between the beta carbon, the effective distance between the beta carbon and the probe is 6 angstroms. And we also constrained the angle, the C alpha C beta spin label angle to be between 120 and 180 degrees, so that there aren't steric clashes between the pseudo spin label and the protein backbone. So using this model, we can now say, given a spin label distance that I've observed for DSL, what is probably going to be the distance between the two beta carbons on the residues? And so we call this the DSL minus DC beta value. So to develop our knowledge base potential, I should say Nathan and Alexander put these the pseudo spin label, randomly placed them on the residue pair in a protein, measured the DSL minus DC beta value, and then put it into that value into a histogram. And he repeated that for the residue pairs in that protein, and then repeated that process for over a database of approximately 3,500 proteins, which included mostly soluble proteins, but also about 100 membrane proteins. And so after he collected this DSL minus DC beta values, he put them in a histogram, which you can see by this line here. And we compare it to actual DSL minus DC beta values for two proteins, alpha A alpha-A crystalline and T for lysozyme. And you can see that the distribution of beta cell lines TC beta values matches up pretty well to the experimental determined value. And then when you, to convert this to a knowledge-based potential or scoring term in Rosetta, we then use the Boltzmann relationship, where basically more statistically favored beta cell lines TC beta values, more often seen values that we've observed in nature are going to be considered more energetically favorable in our modeling, whereas less often observed values are going to be considered more neutral. And so when we're modeling, we'll have a model with a given distance between the beta carbons, and then using the spin label, we can apply a spin label value and then look up the energy of that conformation. How well does it match up with what we've seen in our statistics? So we then took this and optimized the weight of the scoring term and how it was combined with Rosetta other scoring terms. And before we went into the structure prediction phase, we actually tested or compared Rosetta EPR's performance to folding without EPR restraints, and then folding with more promiscuous, less informative, what we call bounded restraint. And we folded 10,000 models just to kind of test it out and compare them to each other. And then to assess the performance, we wanted to see the correlation between the Rosetta energy, and the model accuracy that we obtained for these three de novo folding methods. And you can see that when you, as you change the type of method you use, folding with no restraints to folding with bounded restraints, which is an older method, and then folding with the new knowledge-based potential, the correlation between score and model accuracy, which will be measured by root mean square distance, or RMSD, increases from 0.4 to about 0.5 and then to about 0.6. That's important because ideally you want these plots to more look like a funnel, these more look like blobs, but you would like them to look like a funnel because when you're modeling and you don't know the answer, when you're not testing your method, but rather actually trying to come up with a model to, say, propose more experiments or to support experiments that you've already done, you are probably going to assume that your lower scoring models, the models that agree with your restraints the best, are going to be the correct ones. And so we want this correlation to be as high as possible. So this was a promising result in terms of we can combine EPR data with Rosetta, and that using knowledge-based potential is a good approach. Furthermore, the sampling was improved with the knowledge-based potential, with the Rosetta EPR. So we get more accurate models with Rosetta EPR compared to folding without restraints and with the bounded restraints. And also we get a higher percentage of models that have the correct overall fold. And our cutoff for that is saying that those models must have an RMSD less than 7.5 angstroms relative to the experimental structure. Therefore, we say that those models have the correct overall topology, overall fold. And we also achieved a higher number of models that what we say are native-like, or really quite accurate models that we felt confident we could take on to all out of refinement in Rosetta, which is what we did. We folded 500,000 models of T4 lysozyme with Rosetta EPR, with EPR restraints, 25 EPR restraints, and then filtered them, and then subjected the filtered pool to all-atom refinement, or 8 rounds of all-atom refinement. I should point out that we weren't using EPR restraints for this part, but we started out with good topologies, and therefore, through multiple rounds of refinement, the scores and model accuracy converged. And you can see that the accuracy and the energies of those models are pretty close to the the experimental structure in the Rosetta force field. So here again, I'm putting score Rosetta score as a function of model accuracy, and you can see that they match up they come fairly close to the actual structure. And here I'm plotting the Rosetta energy as a function of rotamer recovery, or rotamer would be what are the conformations of our side chains in the protein? Do we recover amino acid side chain conformations correctly? So turns out that our rotamer recovery increase is better as you go through multiple rounds of refinement as well. And you see that in the end, after our models converged, we were able to recover the correct conformation, even not just the overall fold, but also even side chain in the core of the protein, which is remarkable because we started with the Rosetta folding algorithm, which I showed you the video of, and then only 25 EPR distance data. So, and then we were able to obtain a highly accurate atomic detail model in the end. So that was, folding the T4 lysozyme, a small helical soluble protein. But can we really take this approach and apply it to folding membrane proteins, which is what we're really interested in. Sorry, 13. So before I get into combining EPR data with folding membrane proteins, I'd like to give a brief introduction to Rosetta membrane, which you'll hear me talk about quite a bit in the next few slides. When we talk about Rosetta membrane, it's a special mode in Rosetta where the energy function is specialized for assessing the energetic favorability of membrane protein conformations as opposed to soluble protein conformations. And the energy function has several terms that are specialized just for assessing membrane proteins. Furthermore, Rosetta membrane employs an implicit membrane environment that's subdivided into hydrophobic, interface, and polar regions. And for now, these layers, the widths of these layers are fixed, but I think that might be changing in the future, in the relatively near future. And so not only do we have a set of scoring terms in an implicit membrane environment, Rosetta membrane also employs 4 main energetic penalties that are specific to membrane proteins. We disfavor loop-like or unstructured regions in the membrane, in the implicit membrane region. We also disfavor having N and C termini in the membrane region. Generally, helical membrane proteins sit a little more orthogonal to the membrane plane, so there's a projection, what we call projection penalty, that would disfavor protein conformations where it's more sideways or parallel to the membrane plane. And then also membrane spanning penalty. So the user defines the membrane spanning regions in the protein, and there's a penalty if those spanning regions aren't actually spanning the region. So that's what we mean by, really what we mean by Rosetta membrane. In addition to those penalties in Rosetta membrane scoring terms, we also wanted to add the EPR knowledge-based potential scoring term, like we did for T4 lysozyme. And preliminary testing that I did with Rosetta EPR on membrane proteins, I found that just the knowledge-based potential by itself, where it used to just even out here, did not really help my sampling of correct membrane protein topologies very well. So we added a quadratic penalty here, which instead of less likely, statistically likely DSM minus DC beta values as being considered energetically neutral, we actually now apply an energetic penalty. And this is essentially the same idea as before, where favorable DSL minus DC beta values are going to have lower energy. And we optimize the weight of this scoring term, much like we do with T4 lysozyme as well, but for folding membrane proteins, and combine that with the Rosetta scoring term. In addition to the Rosetta membrane scoring function, there are also, or energy function, there are also different ways of folding or modeling membrane proteins in Rosetta. So you can fold the membrane protein from an extended chain, like in the video I showed earlier, we just start with this long chain and then do fragment insertions. You can do that, but it's hard to sample more non-local interactions that way. One way that Rosetta developers tried to get around that was to apply a more clever folding method, which they call membrane ab initio, excuse me, in which they kind of tried to mimic the two-stage membrane protein folding concept, where the helices are inserted into the membrane first and then come together. The way this works in Rosetta membrane, or membrane ab initio, sorry, is that the user defines, their membrane spanning regions, and then Rosetta picks one of the helices in the middle. So in this instance, helix 4, the fourth helix, and inserts it into the implicit membrane environment, and then performs fragment insertions on that model before inserting the next sequential neighboring helix, and then it does more more fragment insertions, like in the movie that I showed earlier, and then does more helical insertion. And after it reaches the C terminus, it then goes back and folds in the N-terminal helices. And so this is, seems like it could be a better way of sampling non-local interactions, and it's fine for certain proteins, which we'll see in a minute. But one big drawback is that it doesn't, you can't use experimental data with this method for more technical reasons concerning the Rosetta code base. This membrane ab initio folding method doesn't take restraints such as from EPR NMR into account. And so that was a big drawback for us. And what we wanted to do was develop a protein folding protocol for modeling large, complex membrane proteins in which we could also combine input experimental information from EPR. So the past couple of years, I've been working on a protein folding algorithm called Rosetta TMH for transmembrane helix, which kind of combines the two ideas of the traditional de novo folding method in Rosetta with the membrane ab initio method. And I have a movie, but I want to talk you through first because it's kind of a short movie. I first start with an extended idealized helix. And I've removed the loop regions just for clarity, so that you can see the movie better, the protein model better. And then what I do is, as you'll see in a second, I insert all of the helices into a randomized topology, into a randomized arrangement, all at once into the membrane environment. And then treat each helix as a rigid body, and you can move whole helices and translate. You can translate or rotate them, and that for 2,000 cycles, or 2,000 attempts, I should say. And then, after each attempt, you score according to the Rosetta membrane scoring terms that I discussed earlier. And later stages, you'll see there are fragment insertions like you saw before, and those are more to not so much sample protein fold arrangements or the helical arrangements or topology, but rather get more details on the intra-helical features such as bins and cleans. So hopefully the movie plays. So here you saw in the beginning I inserted the helices all at once, and now at first they're doing the rigid body movement, and now we're doing fragment insertions. You see that actually this topology is quite different than the beginning. The beginning topology. Maybe I should play it again actually. But it's kind of nice because you see there's larger movements here, and then now we can try to recover more helical kinks and bends and stretches. So, I wanted to test Rosetta TMH on compare it to the other Rosetta membrane protein folding methods, and we want to test this and assess its abilities on a range of proteins. So I benchmarked Rosetta TMH on 34 membrane proteins, helical membrane proteins, a subset of 11 of which I've shown here. Basically, what I'm showing is that the range in size from about 90 to about 400 to 500 residues, and also in topology or complexity. I divided the proteins in the benchmark set in terms of size categories. So I have small membrane proteins, medium membrane proteins, large membrane proteins, and very large membrane proteins, which is the size category is a combination of how many amino acids each protein has and how many the complexity of the topology. Also, we simulated EPR restraints for our benchmark of Rosetta TMH because there are relatively few membrane proteins for which there are experimental structures available and EPR data available and that I could actually apply Rosetta TMH to. And so we simulated our EPR restraints, and the number of restraints chosen was 20% of the number of the amino acids in secondary structural elements. So, right. So, as I said, we simulated the EPR restraints based on an algorithm that was published by a former Mayo / Machara lab member, Kelly Casimir, who graduated, I think, last year, and using Kraga's algorithm in the in-house software, the Biochemical Library, or BCL. After simulating the restraints, we de novo folded 1,000 models for each of the 34 proteins using membrane ab initio, also folding from an extended chain, and then with the new Rosetta TMH protocol. And when applicable, we also apply the EPR restraints. We assess the model quality by computing the C-alpha RMSD, the normalized RMSD or RMSD 100 over the secondary structure elements, and compare those to compare the model's accuracies to their experimental structures, and then to assess the sampling efficiency of the modeling methods. We determined the percentage of models for each protein that had an RMSD 100 of less than 8 angstroms relative to the experimental structures. This is the for membrane proteins to cutoff we use to determine it has the correct fold or overall topology. And then later I'll explain that we made RMSD plots by taking the top 5% most accurate models for each. So I wanted to see, for each type of membrane protein, small, medium, large, very large, was there a specific folding method that would be better for one category or another? And so here I'm plotting the mean value for the percentage of models with the correct topology. And what I did actually was for each protein, for each size category, I took that percentage of models with the correct topology, let's say all folded with membrane ab initio, and I took that average for those values. And then I did it for each of the other subsequent folding methods in this protein category. And that's what these bars are indicating here. And there's a couple of main points from this slide, from this video. One is that if you have EPR restraints available, if you can use EPR restraints, they tend to help immensely in your sampling capabilities. You are far more likely to obtain the correct fold when you have experimental restraints, which is not surprising because we are restraining the conformational search space, and so it's a little easier to come up with the correct topology. On the other hand, if you don't have restraints, it looks like it's a toss-up between, sometimes a toss-up between membrane ab initio folding from an extended chain, depending on the size of the protein, and sometimes the complexity of the protein. And lastly, if you have very large proteins, such as 12 helix proteins, for example, the new folding protocol, Rosetta TMH, with the addition of EPR restraints, does a slightly better job of getting a higher percentage of correctly folded models. So it is helping in terms of sampling different topologies for these traditionally very difficult proteins. And then another way to compare two methods to each other is to make RMSD versus RMSD plots, which need a bit of explanation. So imagine that I took the top 5% of models having an RMSD, and by accuracy, and took their RMSD values and plot them on the x-axis for only those folded Rosetta TMH. And then those same protein models, but folded the same protein but modeled with a different method, I took their RMSD values and plotted them on the y-axis of this XY scatter plot. And you can draw a diagonal line across the plot, and what you see is that values that appear above the line, you see values that appear above the line. This indicates that Rosetta TMH is folding better, something better than the other method. Whereas if the values appear below the line, then the other method is performing better than Rosetta TMH. And so I plotted the modeling from several different methods against Rosetta TMH for 7 example proteins. There's a small, medium, large, very large proteins. And the 2 main points I wanted to make with this figure are that the larger proteins, such as rhodopsin, which is green, carnitine transporter, which is yellow, and the nitric oxide reductase, which is in purple, Rosetta TMH tends to help with the sampling of those types of proteins, more so than smaller proteins, like 2BG9, which is 3, I don't remember the exact name, but it's 3 helices. The addition of EPR restraints to Rosetta TMH helps us sample in every case, unsurprisingly. And also, applying the EPR restraints to Rosetta TMH helps us in every case comparing to membrane ratio without restraints. Remember that membrane ratio can't take restraints into account. So that was just another indication that for large proteins, Rosetta TMH might be an option. And further indication of the assistance that we get from EPR restraints is that here I'm showing for 6 example proteins in the benchmark set, the most accurate model folded with Rosetta TMH, but without EPR restraints, and then with EPR restraints. And in gray is the crystal structure, or the experimental structure, and in rainbow is the Rosetta model. And you can see that often, without EPR restraints, with Rosetta, using this method, we can get the correct topology, the overall arrangement of the helices, but by adding in restraints, we often do much better job of getting better packing, better interhelical packing, and overall placement of the helices, and like especially here, in this case. Excuse me. We also sometimes are even able to get correctly the intrahelical features, such as bends and things like that. So, in summary, for this part, I wanted to show you basically what I've learned concerning membrane protein folding in the past 6 years. And I hope maybe this flowchart will be helpful to some of you. And going back to the road not taking idea, I think that de novo folding, you don't really want to take that road if you don't have to, because it's so much more complicated in a way. If you have a membrane protein that you want to model, if there is a sequence homolog that you know of or that you can find, you should definitely go the comparative modeling route. But if there doesn't really seem to be an existing protein structure that you think that you could base your model on, then you can try de novo folding in Rosetta. But the other thing in Rosetta is that we don't model beta proteins, like beta barrels, such as found in bacterial pores and things like that. So if it's a beta protein, forget it, don't use Rosetta. You'll have to find some other method, maybe the PCL. On the other hand, if it is a helical membrane protein, you might have a chance. Is it, but is it, then you have to ask, is it a small protein or is it like relatively large? If it's not so big, if it's less than 200 residues, you probably can get by with folding using the traditional Rosetta folding protocol, folding with an extended chain. Probably. You might have to fold a lot more models than you anticipate, but it might work out. If it's larger than 209 residues, I would seriously consider trying to collect some experimental information. You can. And then the question is, do you have experimental strains? If you don't, I think actually membrane ab initio folding protocol would probably be better for these larger proteins. Although sometimes you might want to use it for smaller proteins if you don't have restraints. If you do have restraints for smaller proteins, definitely use a extended chain method. And finally, if you have a large protein and you have experimental restraints, then you can probably, I think actually Rosetta TMH is pretty promising. So this hopefully will help some of you in determining, which method you will use. It's so confusing, but I think this pretty much sums up the decision-making process. And in summary, I told you two main stories today. I told you about the PR and how we apply that to folding a small soluble protein, T4-lysozyme. And that was just kind of our first step towards developing more complex methods for membrane protein structure determination. What we learned from Rosetta EPR, the Rosetta EPR project, is that we can combine, we can make an EPR knowledge-based potential. We can combine it with Rosetta scoring terms and Rosetta functionality to improve our sampling of native-like topologies for T4-lysozyme. And even we're able to obtain atomic detail models of T4-lysozyme with sparse data sets. And then on the membrane protein side, Rosetta TMH is a novel folding method in Rosetta, and it combines the ideas of fragment-based assembly, like I showed in the video earlier, but also moving whole helices at a time. And so it really kind of expands upon Rosetta's ability to fold membrane proteins in terms of now we can model even more complicated membrane proteins than we could before. And that's important because now we might be able to use Rosetta TMH as a tool to, especially if you have EPR restraints, perhaps you could use the modeling to kind of come up with an initial model for hypothesis generation and perhaps to help you propose new experiments to then go on an iterative process of modeling and then proposing new experiments and modeling, etc. Which is a method that we employ in our lab quite a lot. So with that, I'd like to thank everyone ever. So firstly, there's a lot of people to thank. Firstly, I'd like to thank my PI, Jens Myler. Like he said, I'm pretty sure I am at Vanderbilt just because of Jens, because he was the only person that interviewed me for the CPD program. It was quite an interesting conversation. And I'm really excited that I got to do my PhD training here at Vanderbilt. Also, Nathan Alexander, he was my mentor in the my lab when I first rotated, and he's been a really great go-to person in terms of resources, in terms of trying to talk scientific concepts out. He really was a great help, and he also did the cone model statistics in the Rosetta EPR project. I'd also like to thank Sam DeLuca, because when I came to the lab, even though I had been doing computational chemistry, I didn't have any experience with scripting or programming. So basically, I think Sam taught me how to do all of that. So introduced me to Python and databases and all that stuff. So it's been really, really useful. Brian Miner was a postdoc in our lab. I'm really grateful to him for providing the input files for the benchmarking of Rosetta TMH on membrane proteins. Also Axel Fisher, because he basically answers all of my questions really quickly on how to use the BCL, especially for simulating EPR restraints. So he's been massively helpful. And also just in general, my colleagues in the Miner lab, they're a great support. They're great people to work with. They answer all my random questions, and it's a really fun place to be. Earlier on in the Rosetta EPR project, we were collaborating with the Masharov lab. Hassan Masharov and Kelly Casimir were really great to talk to, get their input on incorporating the EPR information into Rosetta and how that informs our modeling and what are the drawbacks, and great discussions there. I'd also like to thank collaborators in Leipzig, in the Bexinger laboratory and the Houston laboratory. We collaborated on 2 projects concerning modeling of peptide GPCRs, which I didn't even get to talk about today, but are part of that are part of my thesis work. And they are really also great to work with, very easy to work with. Also like to thank my thesis committee, who have been massively patient over the past 6 years, and I feel like I can go to them anytime when I'm having trouble, and they've been really good for guidance. Also, the CSB and ACRE administrators, Veronica McLean is no longer here, but she was pretty awesome in terms of helping me get all the bureaucracy stuff that I need to get done. And none of the research would have been possible without the ACRE administrators. I lost count of how many trouble I filed over the years. Also, in the CPB program, I'm in an interesting, unique PhD program, the CPB program. And we have great leadership with having Hassan Mashar and Bruce Damon at the top, and then Lindsay Myers. I really have no idea where our program would be without Lindsay. I feel like she's the glue that holds us all together. She's a really great administrator, and it's been awesome to work with her over the years. I'd also like to thank the Red Office counselors, Misty especially. She has been really essential in me getting to where I am today. And also Kim Petrie, because she's in charge of the Red Office career development, and I got my current ACS fellowship probably because of advice that I got from her. Also, I'd like to thank the Rosetta Commons. Rosetta is kind of a beast to work with, and so we have a very nice community of developers and users that we all help each other out when we're having trouble. And then finally, also to thank my NIH funding. I guess that means actually, ultimately, I'll be thanking you, the taxpayer, for funding this research. And then on a more personal note, you know, I wouldn't be here without my friends. And, a lot of them come from lab, actually. So me and Liz and Nathan with his peanut butter and jelly sandwich. This is how I remember our office all the time. 5154G was a really great place to be working. Also, Stephen has been great for all kinds of interesting discussions over the years, and ranging from science and not science. Also, my eating out buddies, Yen and Amanda, and you know, Kate is not in our lab, but she's in the CSB, and I've had the privilege of getting to know her more over the past few months, and she's been a really great support. And my friends from college, Tian and Ed, I don't really talk to them very much, but they're always there. They're always thinking about me. I'm happy to still be friends with them. My friends in the NFB, the National Federation of the Blind, are really good at they're really good at empowering you and making you feel like you can do whatever you want, and the blindness can just become an inconvenience, more like. And they're definitely a no excuses group, and they definitely, you know, are very encouraging whenever you set your mind whenever you set your mind to do something. And my girlfriends in Nashville have really been great for keeping me well balanced, especially when I need to get time away from Sam because we spend so much time together in the same lab. My friends in Nashville IPCAI. This is a great place to do Aikido. I love martial arts. I didn't get to go nearly as often as I would like. They're not just a great place to train, but it's a great group of people to get to know. And I will miss them sorely. And my family, you know, I'm really glad that both my parents couldn't make it today. My sister and my grandmother. You know, I was adopted back in the 80s, and you know, I can't even think of a better family to be raised in. So here's a picture at the wedding of my mom, my dad, my sister, and my brother who couldn't make it today. And Brian has 2 sons, and then my other brother, Isaac, has 3 sons. So my 5 nephews and my sister are really kind of I see them as a great reason to try to set a good example, a very good role model, and they really inspire me every day. I'm also really fortunate to have the best in-laws that anyone could ask for. They're like the opposite of my family, but we get along great. So I think that's actually a blessing. Marie unfortunately couldn't make it, but Alice and Ed are here. And I feel like I've been adopted by them too. And then finally, there's Sam, who, excuse me, I don't know. We do a lot of trading the same advice back and forth over the years because we're in the same lab and the same program. But he's really good, been there for me and been a good sounding board and been really encouraging and supportive. And he likes the same things that I like, so that's great for me. He loves to travel like me, so he's a great traveling buddy. He also is really good cosplay Doctor Who characters for me. Driving cons. And he loves cats, which is super, super important. So, and you know, I have to thank my cat because she's really a good therapy animal, and she's really I miss her as much as I miss them sometimes. So I think that's it. I think that's all the acknowledgments. I'm sure there's many more people that I need to thank, but I can't think of them right now. But if you have any questions, I'd be happy to entertain them.
So we take questions from the general audience at the time. And the committee, please.
I think if you have questions, maybe you should just kind of shout them out or tell me who you are. I can't tell if you're raising your hand or not. So.
Stephanie, I have a question. This is Kate. So I know when you have deer data, or at least I think when I've seen deer data, you typically don't get distance restraints. You don't just get like, oh, this is, 10, 20, whatever angstroms away. You get a distribution of distances, right? Yeah. So my question is, so you have this, bell curve, and I know you have this like cone model that's implicit in your program, but I was wondering if, these bell curves, they can also be, the distributions can also be influenced by the dynamics. So when you simulated your distance restraints, did you simulate this distribution, and is there a way to take that into account?
Well, so there's 2 types of distribution that you can get. Well, that's probably the representation, but your distribution can be based on actual dynamics of the protein, or it can be based on the actual movements of the spin. It's probably both, but in the code model, right now we don't really account for the overall ensemble of molecules that you get that you would often observe with deer, but the code model and the statistics we're going that do kind of take into account the the dynamics of the technical stuff. And you saw the distribution is very broad, and that's because it's taking that into account. One of the future directions that I think would be really cool to do is, and now that Mosaic has worked with MySQL databases, that you could do an iterative process of de novo folding, and then filtering out for models that meet those larger probability distribution POC. So you can end up with an ensemble of models that actually fits the probability distribution. Right now we don't do that, but and I mean one possibility is to do post processing thing too, but I haven't done that yet.
This is not a defense unless you guys attack.
I think they're just saving it for you.
Is there any, this is Rocco, by the way, are there any things that you might, if you had more time, that you might like to add to Rosetta TMH to potentially improve the performance?
Oh, there's so many. Science is infinite in method development in that way. So, like I was just telling, like I was just mentioning, there might be one thing that would be cool is generating ensembles of models that match the EPR distributions. One thing that I noticed in this last round of analysis is that actually we really need to implement a loop constraint, loop closure constraint, and a loop knowledge-based potential, much as what is seen in the BCO. There were plans to do that. There were ideas. There just wasn't enough time. But that is definitely one thing that Rosetta TMH needs, is some way of constraining how far the helices can move from each other. Right now, the EPR data do that, but in the absence of EPR data, the helices tend to just move on afterwards. So a loop knowledge-based potential loop constraint. Also, there was also a possibility of implementing long EPR accessibility information into Rosetta. That would be really helpful for membrane protein structure determination, because that would help us to get better orientation and placement of the helices based on accessibility information obtained from EPR. And the list, it could go on for a while, and we can talk about it later if you would like.
Thanks.
Last chance. Okay, then before we thank Stephanie again, I just wanted to say, and then ask all of the guests to leave the room and the committee stay here for the session. Thank you, Stephanie.